Okay, so we're going to pick back up with part two of urinary system anatomy. And so we're continuing on with the nephron. And this, I can't stress it enough how tiny these things are. So one time, um, this has been like a few years ago, we were in lab and we were looking at the kidneys in cadaver lab. And I had a student, after we were done, we came back over to the classroom and he was like, we didn't even see the nephron. I was like, well, nephrons are tiny. They're microscopic. If you think about that kidney, that's about the size of a bar of soap, and there's a million of these in there. So they are really, really tiny, very microscopic structures. So one of the things that we talked about before is how the nephron consists of both the renal corpuscle, which then it consists of the glomerulus, which is the capillary, and the Bowman's capsule. And then the nephron, the second part of the nephron is the renal tubule. And so that's what we're talking about here. And so basically what we have is if we kind of draw this out, we've got here's our glomerulus, so here's our tangled mess of capillary, and we've got blood heading into it through an afferent arteriole, and blood heading away from that glomerulus through an efferent arteriole. Now surrounding that is our Bowman's capsule. And so that's going to basically look like this, right? And our direction of filtration, so we've got fluid moving from the glomerulus into Bowman's capsule, and then it's going to start to travel through this tubular system. And we've got different parts of the renal tubule. So the first part is going to be the proximal convoluted tubule. And so proximal is basically its relationship to Bowman's capsule. So the first part of this tubule is going to be here, and we call this the proximal convoluted tubule. And then we're going to go through this nephron loop. And this is also called the loop of Henle. And so it's basically going to have a descending part that's going to travel down, and then an ascending limb that's going to travel back up the other side. And so here's the descending limb, and here's the ascending limb coming back up the other side. And then we end up with a distal convoluted tubule here. Okay, so this filtrate is going from the glomerulus into the proximal convoluted tubule. It's going to go down the descending limb of the nephron loop. It's going to go back up the ascending limb of the nephron loop, and then it's going to go out the distal convoluted tubule. And so this is all part of the renal tubule. Okay? And so this is what you see here. This is basically what we just drew. So here's our renal corpuscle with the glomerulus and then Bowman's capsule around it. Here's our proximal convoluted tubule. Here's our descending limb of the nephron loop. Here's our ascending limb coming back up the other side. Here's our distal convoluted tubule. And then we're basically going to drain out of the pyramid from there. But another thing to look at too is that we'll talk about this. These guys, these structures are what give the renal pyramids their stripes. Basically, it creates the striations that we see in that tissue. And generally, what we see is that the proximal part, or the renal corpuscle, starts in the cortex of the kidney. So it's kind of along that outer edge, and then these tubules kind of dip in further and travel down into the pyramids of the kidney. So just a couple details about these. When we head into proximal convoluted tubule, it's kind of a hotbed for reabsorption. And so what we see there is adaptations that allow that to happen. So the apical surface, which is basically, this is looking at it in cross section. This is going to be the lumen of the tubule here. What we see is this brush border all in through here, and you can see it in here too. This is on the apical surface of these cuboidal epithelium, and basically that creates a brush border of microvilli to increase surface area. And we know that when we have structures like that present, it's because we're increasing absorption in that area, and that's exactly what's happening here. So that's a great example of complementarity. Through the nephron loop, so as we dip down through the nephron loop, through the descending limb, typically what we see is the proximal part of the descending limb is thick and continues with a cuboidal epithelium similar to the proximal convoluted tubule. And then what happens is it basically gets thin. It's hard to draw it thin, but so it basically becomes a thinner loop and is made of simple squamous epithelium. And then as we head back up the ascending part, it's also going to begin as thin, a simple cuboidal or columnar, and then get thicker as we head up. So this is kind of direction of flow here. This is going to come into play when we talk about physiology and what exactly is happening here. But realizing that in the nephron loop, we've got some parts that are considered thick and other parts that are considered to be thin. And so that's just another highlight of what we're talking about. And then as we head into the distal convoluted tubule, what we have here are its cuboidal epithelium and very few microvilli. So we don't really see the brush border. It's kind of a smooth apical surface. So if you had to guess, you would say, because you're not seeing that microvilli, that not as much reabsorption is happening in the distal convoluted tubule when compared to the proximal convoluted tubule. A couple different ways to look at that, and it walks you through the different types of cells. So what happens with these is that the renal tubules are going to drain into collecting ducts. And basically the nephron ends at the distal distal convoluted tubule. So we're going to have, the Bowman's capsule, we've got proximal convoluted tubule, we've got our nephron loop, we've got distal convoluted tubule, and then what's going to happen is that that distal convoluted tubule is going to drain into a collecting duct. But what ends up happening is that many nephrons drain into each collecting duct. So maybe we have one coming from this side, and maybe we have another one coming from here. So because we have many nephrons draining into each collecting duct, the collecting duct itself is not part of the nephron because lots of nephrons drain into it. So it's those collecting ducts that you see that run through the pyramids. So if you think back about the medullary pyramids that we had in the renal medulla, right? They were kind of triangular shaped structures, and we talked about them being striped. And the reason that they're striped is because of these collecting ducts. So they're going to run through them, and they're all basically angled towards that apex and heading down that direction because we're going to drain fluid. It starts as filtrate and becomes urine, and it's going to drain through the collecting duct, so here, and out that medullary pyramid. Okay, and so this is what we see here too. Here's all your collecting ducts draining towards, this is the apex of the pyramid, creating that papilla, and then kind of this cup-like structure underneath, which is the minor calyx. So basically everything's heading down, in this direction. All those collecting ducts are draining urine towards the apex of the pyramid. When we look at nephrons, there are two types of nephrons. that we can talk about. So we can divide nephrons up into cortical nephrons, and this is most nephrons. So like 85% of nephrons are cortical nephrons. And if you think about what that means, it means that they are primarily located in the cortex of the kidney. So they are going to be out in this, area here, that outer third of the cortex of the kidney. And then the second type of nephron is called a juxtamedullary nephron. And these guys are kind of interesting because they have these really long nephron loops that dip way down into the medulla of the kidney. So they're going to dive really, really deep. And these guys are most important in creating concentrated urine, which is really important for us because we don't want to lose all of our water in our body through urine. So we want to save the water as much as we can, and we want to concentrate our urine so that we're just getting rid of things that we don't need, and we are holding on to or reabsorbing the water as much as possible. So, but only about 15% of our nephrons are juxtamedullary nephrons. So these guys would account for about 15% of nephrons. Over on the other side, we've got cortical nephrons, and these guys are the grand majority. They're more like 85% of nephrons. So if you look, here's on this side, here's our cortical nephron. And if we kind of take this line and extend it, from here up, this is the cortex. And from here down, this is the medulla of the kidney. And so you can see this little guy here, this little cortical nephron, it's basically in the cortex. That little nephron loop is a lot shorter, and it's not quite as long. If you look on this side, this is our juxtamedullary nephron. So look at this really, really long loop of Henle. And we've got a couple things here too. We've got a capillary network that's going to surround that loop of Henle, and that is called the vasa recta, and that is specific to juxtamedullary nephrons. So that is a capillary network. So anything that's going from the tubule back into the capillary is happening in that long loop of Henle, and is being reabsorbed by those capillaries, and those are specifically called the vasa recta. Okay, so there are two types of capillary beds in nephrons, and we've talked a little bit about this, but let's just tie it all in together. We've got the first type of capillary bed is the glomerulus capillary bed, and this is that tangled knot of capillaries, and we keep kind of drawing it like this, but this is basically what it looks like. There's our tangled knot of capillaries, and it's got arterioles that are leading into it and out of it, which is also a little unusual because typically a venule drains a capillary bed. But in this case, we've got arterioles that do both. So we've got the afferent arteriole, which is bringing blood to the glomerulus. So that would be, let's say, on this side. And then we've got the efferent arteriole that's draining blood away from the glomerulus. And we'll say that's on this side. Now, another thing that's unusual about this is that the glomerulus has really high pressure. And so think about that for a second. When we did blood vessels before, what was one of the things we talked about in terms of pressure changes that we see throughout the circulatory system? What we typically would see was capillary beds have very low pressure. So if you guys remember, this was kind of we drew this chart out a lot. Like here's our pathway, here's the aorta, here's capillaries, and then here's like on the venous side of things. And our pressure is highest in the aorta, right? And then it's going to fall, and it's pretty low in the capillary beds, and then it might come up just a little bit on the venous side of things. But if you look at our pressure curve here, it's very low. Typically, capillaries have very low pressure, but the glomerulus is different. It's actually got high pressure because we have the process of filtration going on here, and we've got to filter all of that fluid that passes through our kidneys. So the efferent arteriole is going to lead out of the glomerulus, and it is going to create a second capillary bed. So here's our second capillary bed. That's called the peritubular capillaries. And so these are the capillaries that surround the renal tubule, and specifically the nephron loop. Okay, so these are the capillaries that are responsible for reabsorbing most of the filtrate that's moving from the tubule back into the capillary. And these serve as sites of reabsorption. And like we said before, the vasa recta, that is a type of peritubular capillary, but it's the type that's specific to those juxtamedullary nephrons with those really, really long nephron loops. So this is what we see here. Here's our first capillary bed. This is the glomerulus. Here's our, on this picture, we've got afferent arteriole leading in this way. Efferent arteriole is always leading away from the glomerulus, so it's going to be indicating this end. There they are again. Little, see, right? Little tangled knots of capillaries. That's what that looks like. And then this is highlighting the peritubular capillaries. So on your little short cortical nephrons, here's your peritubular capillary. So efferent arteriole is going to leave the glomerulus and create this peritubular capillary network. On our juxtamedullary nephrons, we have efferent arteriole that's going to leave the glomerulus, and it's going to create this very kind of complicated capillary network that's surrounding that very long nephron loop, and that's specifically called vasa recta. Okay, we do have this another kind of interesting structure associated with the capsule and the nephron, and this is called the Juxtaglomerular Complex. Okay, and so we've got two different types of cells that are here, and so where this is located is what ends up happening with nephrons is a lot of times we like to draw it out. We like to draw it very linearly, and just like I've been drawing it, where we've basically got our glomerulus, and we've got our capsule, and we've got our proximal convoluted tubule, nephron loop, distal convoluted tubule, heading out that way. Nephrons aren't really arranged like that. That's not really how they're arranged. They're actually arranged so more like this picture here. Okay, so in this image, we've got proximal convoluted tubule here, and descending limb of the loop of Henle, ascending limb of the loop of Henle, or the nephron loop coming back up the other side, and then we're going to start into distal convoluted tubule. So you can see like the nephron's kind of folded on itself. It's not all linear like we tend to draw it out, but what we end up creating is this very interesting area here, and this is what's called the Juxtaglomerular Complex. And so we've highlighted this area and we've kind of blown it up. So that's what we're seeing here. Here's the afferent arteriole leading into the glomerulus. Here's the efferent arteriole leading away from the glomerulus. And if we've done a cross section kind of through this area here, we can pick up on this structure here, which is going to be the ascending limb of the loop of Henle. And so that's what we're seeing here. But where all of these structures come together creates a very special area. And we've got two specialized types of cells in that area. One of them is called macula densa cells. And so basically these are located in the ascending limb of the nephron loop. And so maculodensa cells are very specific types of cells. They are chemoreceptors, and they are measuring the amount of sodium chloride in the filtrate. And that's going to tell the kidney how quickly fluid is moving through, because if fluid is moving through very quickly, then the nephron loop doesn't have time to reabsorb all the sodium chloride. If the fluid is moving slowly, then the renal tubule has more time to reabsorb sodium chloride, and the sodium chloride concentration in the filtrate is going to be lower. So it's able to monitor that through chemoreceptors. So if we look here, that's going to be these cells here specifically. So if you think about it, it kind of makes sense that they're located in the walls of the ascending limb of the loop of Henle, because they're measuring content of ions in the filtrate. They need to have access to the filtrate, so they're going to be in the tubule. Okay, okay, keep going the wrong way. The second type of cell that's located here in the juxtaglomerular complex are granular cells. And so these guys are responsible for secreting renin, which is an enzyme, and it begins this whole cascade of renin to angiotensin 1, angiotensin 2. But the main point of this is the ability to monitor and regulate blood pressure. And so where we see these granular cells is because their blood pressure, they are going to be mechanoreceptors, because we know mechanoreceptors are going to respond to mechanical events like stretch and pressure, and they're going to be located in the arterial wall of that afferent arteriole. So if we look here, here's our afferent arteriole leading into the glomerulus, and if you look on the side, you've got all of these little cells that are on the side here. Those are the granular cells, and so they're going to measure, kind of constantly measuring blood pressure leading into the glomerulus. Okay, so that's just kind of an introduction to that juxtaglomerular complex. We're going to get a little more into that when we go through the physiology.